invite our next speaker, Professor Boris Martinac, to share with us his work.
Yeah, thank you.
Thank you, Hanjay.
Okay, hello everyone on any side of the world. So good morning, good evening, and good day. So I am Boris Martinac. I'm head of the Mechanosensory Biophysics Laboratory, Victor Chang Cardiac Research Institute. And one of, as Joshua mentioned, one of the major attraction for me to participate in this symposium is that Joshua promised me to keep a spot for me on the first rocket to Mars. So I don't know if they are going to accept me because I'm a bit overweight. But anyway, let me share with you the my screen now. So this is here, and I will do this. Okay. Share. Okay. Am I sharing it with you? Should be. Okay. Can you see my screen now?
Yes, we can see the screen. Thank you.
Okay, great. Okay, so I'll be giving a very actually brief overview and not being able to touch on anything on mechanosensing and mechanosensing.
Oh,
sorry.
I think your presentation is it has your notes on it.
Okay, that's fine. I can switch then quickly, and this is display setting is show presenters. Okay, good. This is fine. So
perfect.
Right now you can see. So I put the notes on because the video will be there, so people can read. Those who didn't see my talk can then see what I was talking about. Anyway, so the mechanobiology has been really on rise over the last 5 to 10 years. I myself have been working in mechanobiology field for over 30 years and been focusing on mechanosensitive channels, which were briefly mentioned also in previous talk. With regard to mechanical forces, here is shown as an example the vascular endothelial cell, which is has the extracellular matrix, cytoskeleton, and lipid bilayer. Let me just get maybe try to get the laser pointer so you can see better see. So this is has extracellular matrix like all the mammalian cells, cytoskeleton, and of course lipid bilayer, which keeps all these components together. It's like a sandwich structure, and the mechanical, actually the cellular membrane is the major target of mechanical forces, either external or internal. And that's why many receptors have been expressed in the membrane. So the forces are generated either by fluid flow, like in the case of endothelial cells, by blood flow, then cell surface interaction also causes mechanical stress on the cell, and then there are cell-cell junctions where forces arise. And all these forces have been experienced by different types of mechanoreceptors. Among those, I'll be focusing on ion channels as mechanosensors and give you some examples what actually happens when it happens that when ion channels are transmitting the signal and affecting signal pathways. And then specifically will be on mechanosensitive channels, and the channels I'll be talking about are piezo channels, which have recently become one of the major focus of research in mechanosensory transduction, because they play significant role in many mechanosensory processes in not only in humans, but in animals and vertebrates in general. So the pathways which link mechanical force at the cell surface to intracellular signaling and downstream effectors are shown here in a cartoon form. On the left side, we have types of mechanical stress like shear flow and cell adhesion, which cause the force being exerted on the cell. And as I mentioned, plasma membrane is a major target, and plasma membrane contains many mechanosensors, including cytoskeletal adherins or integrins and integral membrane proteins like mechanosensitive channels. These mechanosensors are at the very forefront of the signaling pathways within cells. And then if they are attached to cytoskeleton, they basically transmit the mechanical signal all the way to the nucleus, where the gene expression is affected, so that at the end, the cell differentiate differently, adapt to their differentiation to the external mechanical environment. They migrate according to that, and then all this affects their survival, as well as the suicide processes known as apoptosis. Now, briefly about piezo mechanosensitive channels. They have been discovered back in 2010, just 10 years ago. And they are extremely important because I'm just showing a couple of the mechanosensory physiological processes where they play a role. It's in hearing, they seem to be evolved as well. Then physiology of bone, they play important role next to TRPV4 channels, which I mentioned in the previous talk. And then as well as in a mechanoelectric feedback of the heart, we have been working also on a project where the piezo channel play a role in the mechanoelectric, cardiac mechanoelectric feedback. Also then piezo 2 is very important in the sense of touch. And as well as when you feel an urge that you have to go to the toilet, also piezo 1 is signaling to your brain that it's time to go there. So the reason I put also example of bacterium E. coli is that these channels in E. coli exist to serve as osmotic valves to regulate the osmotic pressure in these cells. And the reason I'm showing that is because the physicochemical principles which govern the functioning of these channels in bacteria apply all the way to piezo channels and many other, if not most, if not probably all mechanosensitive channels known today. So this is basically, it says that nature once found a way that something works. Basic principle, if it works, don't fix it. So it keeps going on because evolution allows for that. So now piezo channels are widely expressed mainly in hollow organs, large expression in stomach, endothelium, blood vessels, lung, and bladder. Although, as I said, they are also to some extent expressed practically in almost all cells in the body. Thanks to the cryo-electron microscopy, which is another revolution in structural studies of proteins, and particularly membrane proteins, which were hardly accessible to crystallography. These channels have been now, structure of piezo channels has been of both, actually. Piezo 1 and Piezo 2 has been solved by using cryo-electron microscopy. And here is shown Piezo 1, and it is has a form of triskelion, like this old symbol going back thousands of years. And then, as you can see here, you have one monomer and 3 of such monomers, one monomer emphasized in blue, and 3 of such monomers form this triskelion. And it's one of the largest proteins known. It's about 2 megadalton in molecular weight. And once they embed in the lipid bilayer of the membrane, they basically curve the membrane. They cause the curvature as according to the current evidence. And the way they are operated, when tension is applied, then the channel is stretched, and the stretching of applying the stretch on these blades of the propeller and pushes the beam against the pore region, and that channel opens and lets the ion through. What is really important is that these channels are selective for calcium, and as probably everyone knows, calcium 1 is the most important second messenger in the physiology of living in mammalian cells, for example, and they trigger many enzymes that are dependent on the calcium, intracellular calcium concentration. So what we have studied in our laboratory, we looked at how does expression of piezo channels affect the content of the lipids in the membrane bilayer. And what we have found is, it's really very striking. We use the mass spectroscopy to do lipidomics analysis. And we use 3 types of cell, wild types. Then we have cells of which, in which the channel has been knocked out. And then 2 types of overexpressing piezo, a little bit higher overexpression, and then very high overexpression. And then we looked how does the membrane environment changes based on the expression of piezo channel. And it's we found that a large number of genes shown here. I'm not going to go into detail. This will be, we are preparing a publication on that. This will be published hopefully in the near future. But basically a large number of genes are upregulated and downregulated depending on the level of expression. That's why we have 2 levels, because we can see differences in expression of different genes. And so in summary slide, it shows that piezo curves cell membrane and surround itself by lipids which favor its function. And then when the channel is either deleted, like in knockout cells, or normally expressed or overexpressed, the lipid content is largely changing in such a way that if the lipids which are important for the channel function are, let's say, affected by some by the overexpression, it may lead to change of function. And these change of function diseases are among the main mechanopathologies linked to mechanochannel pathologies, which have been identified, and usually quite a number of them of genetic origin have been linked to malfunctioning piezo channel, and so on. And this can go, again, goes back on the channel inserting in the membrane surrounding by lipids. And so you have this feedback between expression of channels and affecting environments, and environment affecting the channel. And this is, in this way, this mechanosensory transduction linked to this piezo channel has been regulated within the cells and depend on the forces applied to the membrane. So again, I'm just showing this a little diagram to remind you how mechanical stress via mechanosensors can affect gene expression. So now we come to a new method, which is called acoustic force spectroscopy. Acoustic force spectroscopy is basically the experimental platform allowing you to study the viscoelastic properties of cells by using ultrasound. And why are we using that? Because as I showed in previous slide, expression of channels affects the content of lipids, and this changing of not only lipids, we haven't looked into proteomics, but very likely also the expression of different proteins will be affected to more or less extent, as in previous talk was shown that I think it was vinculin which was affected, and then which is important in mechanotransduction pathways. So what we now are doing, we are looking at these cells and looking how the mechanical properties of cells have been changed by expression, different expression of piezo channels. And what you see here is basically AFS setup in our laboratory here. So you have an inverted microscope, you have these AFS chips, which can be put on this inverted microscope, so you can monitor the cells under the microscope, and then you can apply ultrasound, and this application of ultrasound affects the cells in the way which you can measure their viscoelastic properties. This will become a little bit more clear now in the next slides. So what is happening, this actually chip is equipped with a transparent, very thin piezoelectric element. And this piezoelectric element can be then driven by the voltage, of course, alternating voltage can be driven to reach the optimal resonance frequency, which then generates ultrasounds, and these ultrasounds form standing waves, and one can then, by attaching beads to cells which are tightly coupled to cells, can apply the force, and then once the cells are attached to the bottom, you can basically start stretching the membrane, as I will show you in the next diagram. And the cells can be grown in these small channels, which are about 2 microns in the diameter. And cells are grown at 27, and then temperature is increased to 37 degrees. This is really important because at 37 degrees, cell immediately respond with stretching out filopodia, and they attach to the bottom of the experimental chamber. And one now applies the acoustic force. These beads can be moved up to 20 microns vertically in the chamber. And when we look at the application on the cell, and when cell is attached, then basically beads very similar, like optical tweezers or magnetic tweezers are doing. They basically pull a membrane away, and one can measure then these viscoelastic properties. If the cell was purely elastic, it would be just a single line here. But because of viscoelasticity, this process happens over time. And now we can, from these type of curves, we can determine the elastic modulus and all the other mechanical properties of cell which may be of interest. The great advantage of acoustic force spectroscopy is that instead, not unlike the optical tweezer, magnetic tweezers, one can simultaneously investigate tens to hundreds of cells. Like here shown, you can choose by the program several cells at the same time and pull on them at the same time, and then you get all these number of curves here, and then the average curve among all of these cells is this shown here in black. And these experiments have been done by Giulia Silvani, who was in my lab, and now it's in the Dr. Joshua Cho's laboratory, and Valentin Romanov, who is driving this project as a postdoc here, and my colleague Charles Cox, who is in collaborating together with me, working on these piezo channels. Now, when we look at the effect of overexpression on viscoelastic properties, what we actually see when we compare the wild type overexpressor and knockout, just to make it very, very simple, not going into details of these experiments, you can actually, this paper will be soon published in the journal Small, has been just accepted recently, a couple of days for publication. So in knockout, we don't have any channels. In overexpression, this is by electrophysiology showing their currents flowing upon the mechanical stimulus here. And what we find, find differences in these viscoelastic properties of the cells. They can be determined using the power law model, which links from these material compliance. One can determine then Young's modulus, and as well as the viscous. I'm almost done. And then beta is the exponent which gives information about viscoelastic properties, and you can see that differences in the Young's modulus. Now we come to microgravity, and this is recently started collaboration with Joshua, that he already mentioned what microgravity can do to cells and the body. And what we are going to do, we are going to combine now the microgravity device with the AFS chip, and we will now try to determine also how, let's say, these cells we have investigated for expression of overexpression of piezo channels, how do they respond, how do they, and what do they change, and what does it change in these cells when they are grown using this microgravity device. So I'm very excited to start working with Joshua on these experiments. Now, in conclusion, I'm not going to read that. This will be, if you want, you can read them later on the YouTube or tweet, wherever these talks will be available for next week. And basically, what we have to remember is that gravity, which is omnipresent, like it's basically a force field, which every living organism experiencing is, of course, going to affect, if we change, is going to affect expression of many genes. And it would be very important to find that out, because if we figure out how the mechanical properties of cells are changing, then we can eventually find also the way to support this long travel to Mars and so on, which I hope to be on the rocket. I don't know if I live that long, but anyway, I'm still hopeful. And just to, at the end, to acknowledge my colleagues again, and funding by Australian government from National Health and Medical Research Council and Australian Research Council. And at the end, just briefly, Joshua asked me to tell you something about mechanobiology field in Australia. As around the world, mechanobiology has been really taking off in Australia as well. There are many laboratories, particularly engineering bioengineering laboratories are very interested in this field, because provides the backdoor for engineers to get and do something in biology. And so we recently, in the last year, November 2019, we founded the Australian Society for Mechanobiology. And for those who may be interested in that, our society is next year in October organizing the International Symposium for Mechanobiology in Sydney. And here is the website. You can access and see the details of that. And everyone who is interested is welcome to join us. Thank you very much.
Thank you, Professor Martinac. That was very, very interesting. And hopefully we'll be able to send you to Mars.
Thank you, Joshua. I'm not giving up my hope.
So just to everyone, we have a lot of work to do to be able to fulfill Professor Martinac's dream to go to Mars. In the interest of time, we'll hold off the questions till after the next talk. And feel free to type in your questions if you have anything to ask.